Hi, my name is AlbertLi. I'm Life Sciences, and this is my BlueSky proposal. My BlueSky proposal is about the identification and characterization of chaperone-mediated assembly of telomerase. So first of all, let's talk about what exactly is telomerase. Well, telomerase is a ribonucleoprotein that consists of 4 subunits, 2 of which are polypeptides and 2 RNA subunits. Actually, geneticists have actually identified them in humans as corresponding to the genes TERT, TERC, TP1, and DKC1. Now, the first 2 actually code for the polypeptide domains of our ribonucleotide protein, and the last 2 genes code for the RNA domains of our telomerase protein. So when we consider the folding of proteins in vivo, this is actually quite a stochastic process, because if you consider the size of the cell and the concentrations of these subunits in the cytoplasm, they're actually very low actually. In cells, there's a mechanism in which chaperone proteins or helper proteins will come and assist the folding or the aggregation of these subunits into a functional protein. Although scientists in the past have actually identified several classes of chaperones that assist the folding of telomerase in yeast cells, specifically proteins related to the HSP90 family of proteins, or heat shock proteins. They haven't found sort of the homologs of these chaperones that assist in telomerase assembly in humans. So that's what our project is going to focus on: the identification of the molecular chaperones involved in telomerase assembly. So the first step in our search for these chaperones involves a screening for all possible chaperones that exist in a cell. So we're going to need to actually use a cell line, a human cell line, to conduct the study. Our cell line of choice is going to be a HeLa cell, which is a well-characterized human cell line. And we're going to actually modify our HeLa cell line by placing each of these 4 telomerase subunit genes under the control of a universal promoter. We know that these 4 subunit genes are actually located on distal chromosomes, so we probably need 4 different promoter sites. And so we can place them under the control of the same promoter through site-specific transgenesis. Our genetic construct for one of our transgenic HeLa cell lines is going to look something like this. Here is our gene of interest. Let's just say this is going to be TERT. And we're going to place this under the control of some promoter that we know that has ubiquitous universal expression, and that is, more importantly, inducible. One of these promoters that I would suggest is a tamoxifen induced promoter. What we can do in the first step for our screen is that we'll actually upregulate the amount of telomerase expressed in our HeLa cells. That's going to be one control study, and we can do that because we've placed each of these subunit genes under control of a universal promoter. And in our second group of cells that we're going to compare the expression levels of telomerase to is just going to be normal HeLa cells. And from here, we're actually going to do a screen for proteins that are overexpressed in the upregulated telomerase cell line. And these are going to be our potential telomerase chaperone candidates. So the second stage of our screen for telomerase chaperones is going to involve the identification of these molecular chaperones from our initial pool of potential candidates. How we're going to do that is actually through fluorescence resonance energy transfer, also known as FRET. So if you have the GFP protein attached to the chaperone, and then you have the one of the telomerase subunits attached to the RFP, then what happens is that when you use fluorescence microscopy, you're going to excite the fluorophore present in GFP, and there's going to be energy transfer between the two binding domains, across the two binding domains of the chaperone and the telomerase subunit, and thus the energy is going to be transferred and it's going to be emitted as fluorescence light of a different wavelength. And from there, when we see this fluorescence energy transfer, we can conclude that there's going to be an interaction. It's likely going to be an interaction between the candidate chaperone and one of our telomerase subunits, and thus we can look at the localization of these interactions within the cell. And sort of as a proofreading mechanism, we can actually knock out our chaperones. And we can do this through the Krelok system. So we can flank our chaperone gene with lots of P sites. And these are just specific DNA base pair repeats that a certain enzyme called Cre recombinase recognizes. And what happens is when Cre recombinase is expressed in the cell, it's going to induce site-specific recombination activity that essentially deletes this gene. So it cuts this section out. And by doing this, we've knocked out the expression of one of the chaperones. And from there, we can look for necessary and sufficient conditions on different permutations of chaperones to see which combination of chaperones are necessary for proper telomerase assembly and function in vivo. Now, all of this experimentation might seem a bit convoluted for just the characterization of one protein called telomerase. So what's significant about it? Well, let's talk about the function of telomerase in cells. So telomerase is ribonucleoprotein, so it has its own RNA template. And in human cells, in living cells, what it does is that it adds nucleotide repeats to the ends of chromosomes. And these nucleotide repeats at the ends of chromosomes are called telomeres. They essentially act as padding for our genome, because what happens during DNA replication, due to the inability of DNA polymerase to polymerize DNA on the lagging strand from the 3' to 5' direction, and what happens is that you get a chunk of DNA that's lost at the ends of the replication forks after each cell cycle, because the mechanism by which Okazaki fragments operate causes the ends to sort of be missing on the new replicated strand. But luckily, cells have the mechanism of telomerase to add padding to the ends of the DNA sequences, and so our chromosome integrity can be maintained as long as our expression of telomerase is healthy. Now, this has implications on both aging and cancer. In the context of aging, what happens is that scientists have found that the lengths of telomeres, or these repeated padded regions of our DNA, sort of dictate how long our cells live, and furthermore, how long we live. And so in individuals who have short telomeres or with impaired telomerase activity, this sort of DNA repair mechanism is impaired, and so these cells tend to die faster, and so lifespan is generally shortened. And so if we can find a way to modulate the expression of chaperones, then we can possibly recover some of the functions of impaired telomerase, for example, or defunct telomerase in human cells. And so this is a new area for gene therapy. Furthermore, in the sense of cancer, so a lot of cancer cells, tumor cells, scientists have found that telomerase activity is hyperactive. So what happens is that these cells are essentially immortal and they keep on dividing because their telomerase lengths are kept really long, and these cells are able to divide out of control. And so again, we can modulate the expression and function of the chaperones in order to indirectly modulate the activity of telomerase. And therefore, we can actually target cells that have hyperactive telomerase expression and actually target these cells and kill them. Because if we can find novel drugs that bind to domains on the chaperones and inhibit their function, then we can inhibit the function of telomerase.